everyone. So my name is Allison Shapic, and my mentor was Dr. Veronica Reha, and my project title was The Effects Seasonal Temperature and Weather Changes Have on Bacterial Growth in Medani University's Pond. Now, the primary goal of this research was to determine those effects that seasonal temperature and weather changes have on the amount of bacterial growth found in the pond. Pond ecosystems are very diverse, and we have lots of different species of animals and bacteria on our macroscopic and microscopic levels. So on the macroscopic level, we have different species of fish, birds, mammals, insects, amphibians, and a variety of different plant species. Some of the pond regulars you can see here on the slide are koi fish and geese. On our microscopic level, we have the most common bacteria found in water, E. coli. And this is so common in ponds because of its entry into the water through fecal waste from mammals, aquatic animals, birds, agriculture, storms, human sewage, heavy flooding, etc. Microbial life is impacted by atmospheric temperature and physiochemical properties of water. And some of those physiochemical properties of water include temperature, pH, depth, and dissolved oxygen and nitrates. And temperature is just one of these factors that we'll be mainly focusing on throughout this experiment. And this research is the beginning of a longitudinal study striving to determine the effects of temperature on bacterial growth and also the relationship between bacteria and different macroinvertebrates found in the watershed behind Medani University. So our purpose of this experiment is to test our hypothesis, which is that as seasonal temperature increases, so will the amount of bacteria and microbes present in Medani University's pond. We want to better understand the relationships between certain types of bacteria and macroinvertebrates, as well as I'm hoping to understand how physiochemical properties of water affect bacterial growth and microbial diversity within pond ecosystems. So our materials used, from left to right, we have a 100 milliliter flask, which we used for sampling water from the pond, a sterile disposable pipette, which was used to transfer our samples onto our nutrient agar plates, and our samples from the sterile water jars onto the plates. Third, we have a sterile disposable spreader, and that is used to spread the culture onto our media, our nutrient agar plates. Then we have our nutrient agar plates, which are used to culture and grow our water and bacteria samples. And then lastly, we have our sterile water jar, which was used in the dilution process during this experiment. So our sampling locations, we have 3 locations from left to right. The first location is location C, cross, then location G, gazebo, and location E, electrical site. Our last image is of the pond water, and all the samples were taken directly across from each different site. And the rocks on the bottom of the pond made it easier for us to go down and collect our water samples. So our methods. So many methods were used during the course of this experiment. We used methods of weather watching, water sampling, dilution, nutrient agar plating, incubation, colony counting, and proper aseptic technique. All of the methods were repeated for 5 sampling sessions, and a total of 15 samples were taken. And all of these samples were taken between October of 2019, January of 2020, within the time range of 11 a.m. to 3 p.m. So first, 3 different sites were sampled from. 100 milliliters of water were taken from all the sites using those 100 milliliter flasks. The samples were taken to the lab for dilution and nutrient agar plating. Now, dilution is its own process, and to begin, we would take 1 milliliter of water with a sterile pipette, transfer that into a sterile water jar and mix it, and then transfer 1 milliliter of that solution onto the nutrient agar plate, and that is a 1 to 100 milliliter dilution. Now, if we wanted to perform a 1 to 10,000 milliliter dilution, we would take 1 milliliter from that 1 to 100 dilution, transfer into a new sterile water jar, mix that, and that would be our 1 to 10,000 milliliter dilution. Now, we only conducted 2 dilutions on our first set of samples simply because we wanted to see how much bacterial growth we would actually get in each type of dilution. And the rest of our sampling, we just did 1 to 100 milliliter dilutions. Then all original and diluted samples were plated and spread. And the original samples were taken directly from those 100 milliliter flasks of pond water. Now, nutrient agar plates were put in an incubator for 24 to 48 hours, and we used that time period to ensure that we would not have overgrowth of our cultures. Then the plates were removed and the bacterial colonies were counted. We used a Sharpie to count the bacteria colonies so we wouldn't lose track of our counts. We had 2 plates used per sample, 2 for our original and 2 for our dilution. So 4 plates were used per location, adding up to a total of 12 plates per sampling session. We also measured the weather and the temperature using the AccuWeather application, just an app on my phone. And we recorded the actual temperature and the real field temperature, as well as the weather. And all my results I put on a spreadsheet so that they were easy to go back and analyze. And both these images are images of colonies found on nutrient agar, and ours looked more like the top picture. Some of them were blobbed, but we didn't actually identify those. So these next 3 slides will be my results in table form. We have the date that the samples were taken, the temperature they were taken at, and the counts of bacteria that we found. So this is table 1 from group C, table 2 from location E, and table 3 from location G. So to calculate our number of counts, which you see in the 3rd column of the table, the averages were used between the 2 original non-diluted samples per location, and only colony counts between 30 and 300 could be used. So for example, if you look at the date, October 25th, 2019, and then you look at our number of counts, we had 111 was our average, and you move that 2 decimal places to the left to get 1.11 times 10 to the second for those 2 places, and then CFU per milliliter, or colony forming units per milliliter, as our units. So the bacterial colony counts from all 3 sampling locations showed an increase in the number of colonies at a temperature range of 39 to 40 degrees Fahrenheit. Our highest location count from C was too numerous to count, our highest from E was too numerous to count, and our highest colony count from G was 1.55 times 10 to the 3rd colony forming units per milliliter. And this proved our hypothesis partially correct because the temperature that bacterial growth chose to increase at was in the middle of the sampling temperature range of 38 to 45 degrees Fahrenheit, not at our highest point of 45 degrees Fahrenheit. Now the lowest temperature did have the lowest bacterial counts from location C and E, which we saw in tables 1 and 2, and a decrease in bacterial colony counts also occurred when the temperature decreased from 40 to 38 degrees Fahrenheit. So the hypothesis that as seasonal temperature increases, so will our amount of bacterial growth. This was hypothesized because of geese found at the pond during the months of summer. And the geese produce lots of feces. So an increased accumulation of geese feces in and around the pond means that there will be more bacteria in the water since feces contain a lot of bacteria. And since this occurred in the summer, that means that we associated the increase in seasonal temperature with an increase in bacterial growth. So the largest growth seen at 39 to 40 degrees Fahrenheit was not at our maximum of 45 degrees Fahrenheit. And this was because the most bacterial growth was not seen at our highest temperature, since bacteria have different optimal temperatures for growth. And biologists categorize bacteria based on different optimal growth temperatures. And these categories include psychrophiles, which have an optimal growth of 0 degrees Celsius or higher with a maximum of 35 degrees Celsius, psychotrophs with a maximum of 15 degrees Celsius, mesophiles with a maximum of 20 to 45 degrees Celsius, thermophiles with an optimal growth temperature of 55 to 65 degrees Celsius, and hyperthermophiles with an optimal temperature of growth at 58 to 113 degrees Celsius. So a decrease in bacterial colony counts occurred when the temperature decreased from 40 to 38 degrees Fahrenheit between our samples 3 and 4. So there was a study done on Legionella bacteria in Quebec that I researched, and they found their spread of Legionella to be in cooling towers in the summer months. And the study revealed that water temperature was a positive significant factor in causing Legionella growth up to 120 days in the future. So this proved that bacteria do thrive in warmer temperatures. So there are multiple different errors that could occur throughout the course of this experiment methods. And some of those were not using a aseptic technique, not removing incubation plates within our 24 to 48 hour period, not collecting in the same locations, not collecting over an extended amount of months, and manual counting rather than using a digital counter. And the evaluation of the hypothesis and results of this experiment provided critical information beneficial to the continuation of this longitudinal study. Now, for pursuing further research, alterations to experimental methods could be made to improve our quality and accuracy of the experiment, some of those being extending those months in which the samples were collected from about midsummer to early spring to get a greater temperature range. We could use meters to measure our physiochemical properties of water. A study in 2018 in China measured temperature, pH, and oxygen levels in urban lakes using meters and found that they had a greater accuracy in their results. We can also conduct microbiological tests to identify types of bacteria collected in the samples. So we can use different types of media like blood agar, as you see in the two top pictures, which is used to identify organisms without knowing their nutritional requirements. These are called fastidious organisms. And we can also use fluid thyloglycate broth, which is seen in the bottom picture. It's pink. And this determines our oxygen requirements for growth of particular organisms. I also wanted to better understand how bacteria are related to macroinvertebrates. And we can do this through several studies that I've researched, which included information about microbes cycling carbon and other natural gases so that they can be used by macroinvertebrates, specifically oxygen and nitrogen. And another relationship between bacteria and macroinvertebrates is symbiosis, or a long-term biological interaction between two organisms. Excuse me. And I read an article about insect bacteria parallel evolution that told us that prokaryote Buchnera and a macroinvertebrate aphid, a type of insect, have that symbiotic relationship in which the prokaryote Buchnera provides aphids with essential nutrients for their survival. So in conclusion, the completion of this research allowed for the discovery of the effects seasonal temperature and weather changes have on bacterial growth. And it was a great gateway for our longitudinal study to compare not only bacteria and temperature, but also bacteria and macroinvertebrates. We are also able to have a greater understanding of how climate change and global warming affect bacterial diversity. And this can happen when different types of bacteria are present in the water. Those could change based on environmental changes like temperature, changing that optimal growth temperature for those organisms who might die off, and new organisms who do have that as their optimal temperature growth may come into that environment. Overall, we had a deeper understanding between the relationships between temperature, bacteria, and temperature, and macroinvertebrates, as well as bacteria and macroinvertebrates. These are my references. And that's the conclusion of my presentation. I hope you all enjoyed. I had a great year with you all in senior seminar. I'd like to thank not only the faculty at Madonna in the biology department, but also my mentor, Dr. Veronica Riha. Thank you.